I request participants to confirm whether my voice is audible.
Yes, sir, it's audible.
Thank you.
Dear participants, respected resource persons, and my dear colleagues. Good afternoon. Welcome to fifth day Faculty Development Program, second session, sponsored by Achar Nagarjuna University, Rusa 2.0, on the title Next Generation Drug Discovery Leveraging AI Tools for Advanced Pharmaceutical Research and Regulatory Innovation. Today afternoon session speaker is Dr. Prahalad Pragalpati, Senior Manager R&D and F&D Baxter's Pharmaceuticals India Private Limited, Ahmedabad, Gujarat. We are the conveners. My name is Dr. Sheikh Mastanamma, Assistant Professor, Achar Nagarjuna University, and Professor K. Tejomurthy Sir, Principal, Anucps. We both are acted as a conveners for the FTP. So one of our faculty member, T. Renu Devi ma'am, is going to introduce the sir. Sir, please. Ma'am, please.
Just a second. Thank you, ma'am, for giving me this opportunity. We are truly honored to have such an esteemed gathering today. This session promises to be insightful and enriching for all of us. Now, it is my privilege to invite our keynote speaker for the session. Before I invite our keynote speaker to deliver the address, let me take a moment to briefly introduce our keynote speaker. Dr. Prahalad Pragalapati is a global R&D leader with over 20 years of experience in oncology and generic drug development, Baxter Pharma. Sir has awarded PhD in the year 2024 under Acharya Nagarjuna University. Sir has a patent of azacitidine formulation, which is a US patent. He has published 4 articles in peer-reviewed journal. He won People Manager Award in the year 2024 Baxter Pharma. SAR has certified Six Sigma Black Belt benchmark and advanced computerized project management. Currently, SAR is working as a global R&D leader in oncology and generic development, Baxter Pharma. We are truly privileged to have such an eminent personality with us today. On behalf of ANUCPS, it is my privilege to invite our keynote speaker, Dr. Prahalad Pragalapati, sir. Thank you.
Thank you for the warm introduction and welcoming me to the complete forum and session. I'm very much honored to be part of this particular session. Thank you. Um, so before starting my session, does anybody wants to have a quick question on what is AA for injectable product development? Because this is a post-lunch session, I would like to check whether my voice is audible or all are awake over there. So I would like to ask, I would like to hear at least one question from anyone. Is my voice is audible?
Yes, sir, clear.
Okay. Let let us start. If anybody have any questions, feel free to ask in between, or you may use the chat box to post the questions, or the alternate option is we have a email ID posted over here, so you can reach out to me as well. Yeah, with this due, I will start my particular session, that is artificial intelligence in injectable product development. So first I would like say the disclaimer, that is whatever data I have been presented over here, that is purely based upon the literature, right? And 1 minute, please. Yes, let me take the pointer. Yeah, thanks. So this complete presentation I have prepared based upon the public literature only. It doesn't have any proprietary or confidential data has been used of the Baxter Pharmaceuticals, and whatever the views, that is purely a personal and academic point of view, right? And this is only for educational purpose only, based on purely literature. So this is the agenda which I'm going to cover today. One is AI market landscape, and next is the foundation concepts of the AI platform in detail, challenges in the injectable product developments, and where AI fits in injectable product development, including case studies, challenges, and 1 minute. Challenges and barriers in future roadmap of the AA, and career in pharma AA. Lastly, the question and answer session. Right? So why we want to see a market, right? We are just in a academic, and we are just educate, we are under a phase of the education. Why we want to see a market? Anybody have any thoughts? Because in the market, what are the boom is there, we have to understand, right? If a student uh, molds his career with respect to current market, that gives a shine to his career also for a long-term growth. So that's what always the market evaluation and market growth we have to carefully evaluate. That's what I have presented over here. So overall, AA pharma market at 2023 was 4.9 billion, which is projected to 45.7 billion by 2020, which means that in a shorter period, in a around a 7 years period, you see the exponential 10x times of the growth. This is very good growth. So this is where we can strengthen our career, right? And so how the pharma firms are using, so around 68% at present the pharma institutes are using this particular AI. So where this AI is using, by 2020 we are saying that 45.7 billion is the expected worth of the AI in the pharma. So where we can see most AI usage in the pharma verticals, the first thing is drug discovery. So why in the drug discovery? I will explain the later on slides also, but because there the cost involves more. So if at this particular area, if you replace with the AI, the molecules can be developed in a lesser time. And next is the formulation development. Next is manufacturing, clinical trials, regulatory, and as well as the supply chain, right? So why we are seeing this growth? Because it's a data explosion, because any data AI can take out and can present within a lesser durations. That's the main path. And there is a regulatory push also. Regulatory is also requesting generic players to use the CTD formats. And as well as the main is cost pressure. Every day the cost of the running organizations are day by day increases because of lot of reasons. And speed to market, in the current stage you might have seen the Zomato, Blinkit. If somebody is getting a goods to home delivery, people will go there only. They will not go to some other supermarkets. The same over here is some organization delivering the patient needs in a quicker, in a innovative way, so the patients will buy that particular products only. So that's always speed to market is much important. So basically there are 6 pillars. I have categorized this particular AI technology landscape in pharma as a 6 pillars powering to next generation injectable development. So how the main usage is a machine learning, right? I will explain you what are the key terms. Main is machine learning, and next is the deep learning, NLP, computer vision, generative AI, and simulation AI. So these are the different types of the I can say different pillars we have to consider for the next generation development. Let us see what is machine learning. Machine learning means it's a predictive formulation modeling, and as well as it's a QSAR, QSPR, proprietary predictions, stability, risk forecasting. All these can be done by machine learning. Generative AI is mainly this is used in the regulatory, in the CTD format writing, and as well as the novel molecule generations, protocol auto drafting. This is where the companies are mainly using this type of the AI. Computer vision, which is used for the visual inspection of the injectable products, wherein the vial, for example, any vial comes, after filling, we don't know whether there is a particulate matter contamination is there, or any microbial contamination is there, or anything. So all those can be detected well by AI technologies. For that, 100% visual inspection can be done, right? And next is the NLP and text mining, which is used for the literature mining for the safety, regulatory query responses, and pharmacovigilance signal detections. Molecular simulations, this is called the in silico API characterization. In silico is nothing but we will do anything on the system. There is no lab is required. Whatever we do by using a system, which is called the in silico characterization. And excipient compatibility screenings and degradation pathway predictions. So all this will be done by this. And pattern process AI, this is a real-time batch monitoring, and NIR or Raman software sensors and continuous process verifications. Let us see, we have heard a lot of terminologies, machine learning and etc. So let us see what all these concepts, because always a foundation is much important before learning anything. So what is AI, what is machine learning, what is predictive learning? So the simple definitions are teaching a computer teaching a computer to perform tasks that normally require human intelligence, recognizing partners, making decisions, understanding language, and learning from the experiences. Right? In this, there are, let us understand each and every keyword. Rule-based AI means what? It follows certain rules, right? But for example, regulatory compliance checklist. So whenever you can see that entering into the airports or etc., they will recognize your face and they will integrate to a Aadhaar, and they will see that whether digitally your face and eyes are matching with Aadhaar or not. If at all matching, then only it will allow, and it will allow the passengers to go inside. That's called a rule based AI. And the machine learnings means these are the learns patterns from the data automatically, no manual rules needed. That more data, the smarter it gets. Means if we have done it 10 batches, 20 batches data, so it will automatically let us know the patterns. Deep learning means it's a multi-layer neutral networks that handle images, spectra, and complex sequences used in visual inspections and as well as alpha folds. The great example for this, you have a IR chromatograph. Just load into this technology. It will automatically interpret for what are the bands are there, whether this compound comprises of the iron or oxygen compound, and what are the stretchings are there. So from this, it will automatically let us know what is that molecule, and as well as the natural language processing. This understood text used to scan regulatory documents, scientific papers, and adverse event reports. Generative AI is creates new content, novel drug molecules, new formulations, regulatory document drafts, powers in silico mechanism. This is where the current generation is more working. So because we don't want any lab, the computer will automatically evaluates whether this particular API is compatible with the defined excipients or not, at which pH range these excipients and molecules are compatible, and if you use this in range, how much shelf life will get. Everything will be given by a generative AI. Let us see what is what is machine learning. Okay, so as we seen that machine learning means, for example, if we have a 500 formulations, okay, and you have a stability results. As I told you, it will predict the stability of the for the new formulation. For example, I done a 10 random trials. From that, we can ask the system, if I do this type of excipient concentration, how a new formulation results can yield. Best example is in pharma. We use this for the solubility. I have did a API plus alcohol 10% I did, 80% I did, and in a combination. You see, always the mono solvent prediction is very easy. When it comes to the multi solvents, it is always a difficulty level is high. Where the difficulty level is high, we have to use this AI to come out with the solutions. And supervised learning, for example, it finds a hidden patterns. Means what? For example, if we do a clinical studies, a 10,000 patient records, and this finds the subgroups, which type of category patients is giving which type of the results. And reinforcement of the learning, here it is like it mainly used in the manufacturing units, where the filling will be done. The vials will comes in the tracks, and from the tracks the vial will be pulled by the robotic arm and will be filled, and again it goes into the packaging. So the complete integrations will be done by this type of the machine learning patterns. Deep layer, and we have just before the slide, we have learned that will give a complex data images. It will learn everything. It will give inference of any type of the data images, right? That is about the mission modeling. And what about the predictive modeling? Predictive modeling means, here we have to, first step is we have to collect the data. Okay, predict means it just predicts, that's all. Okay, first of all we have to collect the data, and then later it organize any errors or any outliers, everything it will categorizes, then it will choose a model. Okay, either it's a, for example, mostly this type of data we use for the clinical data assessments. Okay, like a PVPK, clinical phase 1, phase 2, phase 3 trials, all predictions, this type of if you load all the data in this software, it will predict and it will give the result. How it will be used? For example, some bio study data we have to do. Preliminarily, we'll do the studies in the small number of the patients. The data will be given to the software. Now we have to predict. For example, the drug metabolizes through a liver. Okay, so this set of the patients liver metabolism in this pattern. Hence, for the next level of the study, we have to select the patients who has a liver metabolism rate in this defined pattern, right? Then trains the model, then validate, then predict, and refine continuously. So in this way, this predictive model will work. Let us see how it works for us. For example, particle size. Okay, so it predicts LNP particle size from formulations, compositions, run fewer lab experiments. So LNP means liquid nanoparticles. Okay, so like similarly, so if you did a 10 number of the trials, and now it can predict what if you want X type of the nanoparticles, automatically it will suggest how we can do it. Similarly for the drug release, for example, long acting injectables, this is where industry face a lot of challenge, because the release itself we have to study for the 28 days and even longer. So in these scenarios, this type of predictive models will give a lot of informations. Aggregation risk, mainly this aggregation risk will be there in the proteins and peptides, wherein the pH is most critical. For example, for a formulation, either I have to select a 5.5 pH or a 6.5 pH. We have to pick the best pH before doing any experiment. Okay, so in this way, we can predict which type of the pH we have to take, and etc. Similarly for the pediatric doses, um, without any study, if you have a data of some adults or some any other animals related, it could be able to predict how much dose can be given to the pediatric dose. Stability, generally, we use a Arrhenius equation for that. By using this type of predictive modelings, it can be used very easily, right? So these are the basic models. One is machine learning and predictive models, which commonly used by any of these softwares. Let us see, we have learned about the machine learnings and predictive learnings. Now let us see the AI platforms. What are the different AI platforms that we have, and each AI platform, how we can utilize? So first of all, Schrodinger, right? Most of the people might have heard about the Schrodinger, because this is the top top software which is used by the pharmaceutical organizations in developing the injectable products in a lesser durations. So how it works? This is a physics-based molecule simulations with machine learning to predict how drug molecules interact with the excipients, how protein folds, and whether a drug will dissolve, all before opening a single vial. It is the world leading computerized platform for the injectable drug and biologic development. Very easy thought process over here mentioned. So for example, imagine the testing you have to do a 50 excipient candidates at the atomic level without any lab work. So Schrodinger simulates molecular behavior in 1 hour, showing you which combinations succeed before even you start an experiment. This is called a in situ. So Schrodinger will evaluate what is the molecule, so you have a 50 excipients, which combination and which levels is better for your product. It will give, then only we can go ahead for the lab trials. So here there's a lot of savings, that no need to perform a lab trial. Directly outcome what given by the Schrodinger, you can go ahead and quickly perform that particular trial. A very Schrodinger is much expensive. So how, as a student, how I can use it? That could be your question. Um, just you can download the Schrodinger's academic version. Okay, there are a lot of tutorials are available in the YouTube. Understand how molecules behave at the atomic scale. This is very powerful skill for any formulation development students and scientists. So the key features of this particular Schrodinger is we have it could able to predict the solubilities, molecular binding affinity, and near experimental accuracies. That is very good, and it designed for even if you want to handle a biologics also, this software you can utilize, and as well as simulates the protein foldings and QSPR or QSAR models, predicts physical chemical properties from molecular structure. No lab needed. That is a good with this. So when we see with respect to the R&D scientists at lab, for example, a MAB injection at 150 mg per ml sees early agglomeration. So what they did, they load the protein structure into the bioluminate and they run a MD simulation at pH 5.5, 6.0, and 6.5. The simulation predicts lower aggregation at pH 6.0 with histidine buffer. Only 3 target lab experiments are needed to confirm instead of screening a 15 combinations in the lab, which saves about 4 weeks of work. Again, everything will be counted with the cost. So by using this particular software, you have saved API, lot of excipients, and as well as the time. This is where it gives a benefit. Any questions before going to the next software? Right. So how this pheonics can be used? So this builds mathematical model of the human body to predict how an injectable drug distributes, metabolizes, and clears across diverse patient populations. So before any clinical trials trusted by the FDA, EMA, and every major pharmaceutical company worldwide, they use this particular software. Just think that a flight simulator for your drugs journey through the human body. Input the drugs, properties, and patient types. This software shows exactly where the drug goes, how long it stays, and what does achieve target concentrations without a single blood draw. You can, in very thin line, we can say that you can see the ADME parameters of any drug by using this particular software. Again, so as a students, how we have to use this, the PBPK modeling is a core skill for the clinical pharmacology careers, right? And this particular software offers free introductory webinars on their YouTube channel. Ec- Excellent starting point for the students interest in quantitative pharmacology, right? So the key features of this particular software is it has a virtual population of 1,000 patients. Simulation simulates healthy, elderly, and as well the pediatric and renal impaired. These are the main category of the patients. Generally we will look, everybody knows about the healthy patients. Main challenge will be there for the pediatric populations, how to give a dose and etc etc. So that also can be done by using, and mainly the dose corrections will be done for the renally impaired population. So that also can be done by using these softwares. And the PKPD analysis for the post-study blood sample data. So this also can be done by using this particular softwares, and it predicts the subcutaneous absorption kinetics for the biologics, and as well as the LAI means long-acting injectable formulations. Apart from that, ML assisted model selection exposure predictions, and drug-drug interaction assessments. At a scientist level, how we can use this software is a team developing a monthly SC biologic uses by using this software simulations, 3 injectable volumes, 0.5, 1, and 2 in 1,000 virtual patients before a phase 1 trial. This model predicts 1 ml achieves targets through thorough in 89% of the patients. This data goes directly into the IND filing, justifying dose selection specifically scientifically without running any human dose ranging studies. The good part of all these softwares are US FDA allows using these particular softwares, and they honor the assessments done by you. This is one thing. And another part is risk mitigation is anyhow you will, once this in situ simulations everything done, the practical it will again challenged by using a practical study. So the success of the practical human studies can be increased by using these softwares. You can just observe, just I'm giving a heads up, each and every software will be used in particular phase of the development. For example, Schrodinger, which the before slide what I had explained, we have used at the pre-formulation stage, right? And this particular software we are using for a pharmacokinetic PKPD modeling purpose, such that we can derive a dose, how much I can give, either it could be subcutaneous, intramuscular, or intravenous. So like that. So we have developed a formula, we have understood the solubility, compatibilities, and everything by using one software. We have designed the dose by using this software, and this is the next software where we can optimize the formulation, right? So this is mostly used for the DOE and statistics software for in the product development, and referenced directly in the ICH Q8, Q9, and Q10 guidances. It is used for the AI assisted design to identify the minimum experiments needed to fully understand any formulation to process system. You have to think like this: optimizing a cake recipe with a 5 ingredients, right? Instead of trying every combination, hundreds of experiments, this particular software select 12 strategic runs that reveal everything for injectable formulations. This can save months of the time. You have a DOE software a lot of people may be using by using a mini tab or etc. This is the next level to that. So how students can use? Again, the GMP offers a free academic licenses for the students. You can download it and practice DOE on simple pharmaceutical database. ICHQS Q8 design space knowledge is a highly valued skill in industry interviews for formulation scientists. The key features of this: it generates the minimum experimental set, because how many experiments you increase, that much product knowledge you can gain. But again, there should be some balance is required. With a minimal experimentation, we should get a maximum knowledge. That's what this SAS will help out us. It builds a 3D mathematical maps showing how input variables affect the quality attributes. Apart from that, interactive profiles it will provide which draw sliders to visualize how changing one variable shifts formulation quality in real time. And the last is it also uses non-linear ML models for complex formulations, data beyond classical statistical models, right? As I told you previously, the normal DOI runs, they will just extrapolate, okay, in exponential form, but this is a additional advantage for, with this SAS. How it is utilized in the industry level. So for example, a lyophilized product which has a 5 variables, that is pH, sucrose. pH means it's a pH range, and the sucrose level. PS80 means it's a polysorbate 80, which is one of the excipient, and as well as the protein concentrations and glycine. So there are 5 variables, right? Now by using this software, we can generate a 20 strategic runs instead of a 243 full factorial runs. After testing, the response surface clearly reveals that the pH and sucrose dominates, and the profiler identifies that the optimal formulation is minute, saving an expected 6 months of trial and error basis. So which means that a lot of experimentation has been saved when we follow in the traditional approach. So this software will utilize for a optimization purpose, right? So in a pharmaceutical development, AI has a dedicated softwares which we need to utilize at each phase of the product development, such that scientists can reduce his burden, and he or she can focuses on particularly development mode, right? This is about the next software. This is Citrine Informatics, right? What it does, it guides scientists to the optimal formulation with a number of experiments. It excels at high dimensional spaces such as 10 plus component lipid nanoparticle formulations where traditional DOE cannot reach. This is the next level to the previously we discussed on the SAS software, right? So how we have to think about this? How we can think, that is searching for the buried treasure in a vast field, right? Random digging takes forever, right? So this particular software, Citrine, is a small detector. It analyzes every dig results and directs you to the most promising next spot, converging on the answer in a fraction of the time. This is a very short statement which how this software works can be understand. So again, for the students, so this optimization is a career defining skill in modeling formulation science. You can search this word in the YouTube. This concept appears in a high value industry role for the complex drug delivery system. You can learn this. This will help you a lot when you come to the industry, uh, or when you work in a lipid based formulations which are very expensive. But by using this type of these skills, you can reduce the cost, time, and enhance the skill set. So what are the key features? It recommends the next best experiment after every round of the trial. Model gets smarter with each result. It learns if once we give the feedback, it will go to the next level. Simultaneously optimizes particle size, PDA, encapsulation, efficacy, and viscosity together. It takes a multi objective. Uncertainty maps shows exactly where the model is uncertain, tells you where to invest experiments next time. And as well as it creates a foundation database, builds a searchable institutional knowledge base that persists across projects and scientists. So this particular database not only useful for the one project. If tomorrow some other new project comes, you can utilize this database to that. This software utilizes that database while designing to the next experiment. So this is the how this is utilized in the industry is, for example, mRNA LNP injection is a key 8 key features which are very complex, right? They enter 10 prior experiments in this software. The AI recommends 4 experiments per cycle. After 4 cycles, that is 16 total, the AI identifies the formulation with 94% encapsulation, 120 nm particle size. would, in a traditional process, it required 80 plus experimentation trials. So that is the beauty of this particular software. You can see that every unit, every phase of the product development, we have a particular software. It is not like that for every disease we don't have a same medicine. Similarly, each and every step we have to use a dedicated software to use to come up with a good results. Right? And next is, this is the Simca, and Simca is a pharmaceutical gold standard for the multivariant data analysis. It analyzes complex manufacturing database where hundreds of the sensors fire simultaneously to monitor batch quality in the real time and predict failure before they happen. Reference to the FDA PAT guidance. PAT means process analytical technologies. Let us see how to think. Okay, 200 sensors generating data every second, impossible to watch, right? But this particular software compresses all 200 variables in a simple 2D chart. A green zone means the batch is healthy. Drifting outside the zone triggers an alert. For example, the some filling is happening in a mission. By using this software, by using this software, you can immediately detect how, whether it is in control or not, and immediately it will change the input factors such the filling can sustain in a defined target orientation. So you can say that it's a just a health monitor. So for students, so this particular multivariant data analysis is the core skill behind PAT technologies and real-time real testing. So you can search PCA by Start Quest on the YouTube, right? One of the clearest explanations are available and very relevant to the pharmaceutical manufacturing careers, because in the next generation is with a less manpower, industry wants to run for maximum output. That is called rate of returns. They want to see by more rate of returns, less manpower, higher technologies. Right, and this reduces hundreds of variables and yeah, batch processing instead of the traditional. It's a next level, and it perfects batch accuracies, and also these have a online DSC technologies and radar technologies. Even the batch volume makeups also, it will be controlled by using these radar type of the technologies. So mostly even in the granulations also, this type of technologies they use such that the endpoints can be determined. Next is the um, bench benchling. Okay, this is the electronic lab notebook, R&D based platforms. How these platforms will work, this is very easy. So these are called as a natural languages. Okay, instead of using a lab notebook, electronic lab notebooks will be used. These are very simple versions. This is in silico medicine. Okay, so this is built by the pharma AI, a end-to-end AI platform covering target identification, generative modeling design, and clinical trial predictions. Their AI design a novel drug for the IPF lung disease that reached phase two clinical trials in just 30 months in 2025. They completed the largest Hong Kong biotech IPO. This is about this particular pharma AI. So how we have to think that traditional drug discovery is like searching for a key in a warehouse by trying each key one at a time. But in silico, AI reads all scientific literature and designs brand new keys that have never existed before, targeting exactly the right lock. It designed a novel drug candidate overnight that took traditional methods 5 years to find. Right? For the students, this particular in silico medicine is publicly it can be it's a open resource. It can be publications search their publications in the PubMed. Watch the NVIDIA AI broadcast episodes on In Silico. It clearly explained how generative AI designs drug molecules, a concept increasingly tested in the pharma job interviews, because all industries now they are turning into the AI. So we should know in each every stage which AI can be used, and we should know about their basics. Right? What we have discussed as we have a limited time, so what we have discussed, those are the key features of this. Next is the persist AI. This is very good software, right? So this persist AI combines AI formulation predictions with a robotic cloud lab, because we always, for the Schrodinger also, the first software we have learned, right? In that Schrodinger also, you need a lot of infrastructure. You need a lot of we can say that heavy computer systems in the lab. But these particular predictions with the robotic cloud labs, like our, you can just think like our Google. Uh, Google database, which is not right now, the storage is not in our mobile, right? We'll store it in Google Drives, similar to this, right? Specifically for the long-acting injectables, this particular software is majorly used, and a scientist design experiments on a website and predicts robot Persys robot executes them, testing 700 formulations in 2 months versus the industry standards of 10 to 15 per month. So this is a great software which is useful for development of a very complex drug products. Think that traditionally long-acting injectable formulation development means one scientist, one formulation at a time, 10 per month, right? But Persist AI is a robotic lab that you control from your computer. You can plan the experiments online, robots run them, and the results flow back to you through the cloud. 700 formulations in a month. So this is what about the Persist AI. For the students, long-acting injectables are the fastest growing segments of the injectable market, and monthly injections replacing daily pills, such as the HIV, etc. So Persist AI website has a demo video explaining the platform clearly for the students new to the field. So from there we can learn. And again, these features are thousands of formulations can be done, and we can design multiple experiments and the fastest of the execution, and desired release can be achieved. In the industry, these softwares mainly used for the long-acting injectables, PGLA, uh, PLGA type of the formulations, and etc. And this is Viva Vault AI. So whenever any pharma industry files some dossier with the FDA, this will be done in a CTD format. So all these documents are saved in this particular wallet, and at any time, whether if once we file a dossier to the regulatory, there is some review cycle. Anytime if query comes, you can just you can just go through this software. You can search what in because here anytime filing will happen. How it will happen? Mainly the first filing will happen later on. This is called as in regulatory language, this is called sequence 1, sequence 2, sequence 3. In sequence 1, what is filed? Sequence 2, what is filed? 3, what is filed? There is a very lump sum of documents, but this particular AI makes easy for the scientists. So you can search it randomly, you can search it in a sequence, you can search it by a keywords, anything can be done. So in this way, this wallet will works for the regulatory submissions and keeping those documents searchable and in multiple ways, such that the filing timelines can be drastically reduced. So this is a Medidata Econ AI. This is mainly for the clinical data management platform. Okay, for example, if you have millions of the 6 millions of the patients and 26,000 clinical trials for injectable drugs entering into the phase 1 to phase 3. So this particular software enables smarter trial design, real-time safety monitoring, and as well as the predictive patient analysis. So all this can work smartly by using Econ software, right? So this is mainly for the clinical data management. This is Empower AI. For example, traditionally we develop the methods by using a randomized technologies, but this particular Empower AI will help us in a quick method developments and as well as the validations. Again, as per the FDA requirements, this particular software is 21 CFR Part 1 compliant. So this can be very much useful for the industry. Traditionally, if you want to develop any method, it need at least 2 to 3 weeks for a trial and error basis. But by using this particular AIs, uh, even number of hours, decrease it from the weeks to the number of hours for the method development. So that is a very greatness of this particular Empower AI. So again for the students, these are all open available for the students, and the key features are, for example, you have to. Yeah, any question for me? Okay, so this type of the softwares will give a automatic glycan profiling, peptide mapping, because yep, I don't know, somebody has muted me, so I have unmuted myself. So this is the software which will be used for the analytical method developments in a quicker fashion. And this is the Python or skitlearn, or it also called as a AlphaFold. So this is a this is especially used in the pharma, like buying or pre-built car. You can think like that. You just buy a pre-built car, like that type of the things it is. It means it build custom QSAR models, and as well as the formulation classifiers, stability predictions, and everything can be done by this type of the software. And it is statistically analyze everything, and regulatory biostatistic can be done by using this particular software, and the deep learning and image analysis, and as well as the sequence predictions and generative models, everything can be done by using this type of the softwares. So let us see. So we have learned about the machine learning and as well as the predictive learning. We have learned a lot of softwares are available at each and every phase of the product development, right? So now we learned about the AI. Now we have to integrate to the pharma industry. So let us see what are the different types of the challenges that are there in the pharma industry, such that if I know the softwares, that's what we have learned, and now I have to integrate to the pharma industry. Where I can integrate, we should know the challenges. For example, sterility assurance challenges. Okay, this is they just have categorized those challenges. One is sterility assurance challenges, protein stability challenges, particulate matters challenges, container closure challenges, complex formulation challenges, and as well as the regulatory compliances. So all these are different types of the challenges. So wherein pharma industries generally faces a lot of questions from the regulatory bodies and as well as the market related queries and etc. All these areas we can utilize these particular softwares. So we learn the softwares, we learn the challenges, different types of the challenges. It's a time to integrate. Okay, so which software fits for which type of the development pipeline? So that is what I had did over here. um, this is basically a molecular selections which is done by the marketing teams, and as a pre-formulation studies, we have understood the Schrodinger is a great software where you can simulate the molecular modeling and etc. And next is the formulation design, SAS, like a device software, and analytical. You have seen that the Empower AI and as well as the Water Unify. Next is the scale up and pat tools. This is here it's Persist and Simca. These are the great AI tools generally used. And as a lab data management, which is AI, which is a ELN. ELN is nothing but the electronic lab notebook. And apart from ELN, LIMS will be there. LIMS is nothing but a stability data management. And PKPD softwares, which is generally done by the Phoenix. And next is the regulatory submissions. Regulatory submission is nothing but a software is wallet, like how we keep money in our wallet. All the regulatory data will be kept in this particular wallet. And clinical data management, we have seen that all trial data, safety with data monitoring, everything can be done by using this particular software. And finally, post-market surveillance. So this will be done by Viva Plus Medidata, right? This is integration of the softwares and where we can use at a different phase of the development. Let us see a real world AI case study in the injectable product development. So that is for example, mainly industry faces a development challenge in the protein biologics, where you see the aggregation of the biologic formulations, because these are the large molecules. In this scenario, AI solution will be the ML model using a 120 excipients parameters to predict aggregation risks. So in this way we can mitigate the protein biologic product developments. In this, again, it's a very by this time we can know all the benefits, that is only fewer stability failures, formulation timeline will reduce, and lot of money saving. In the lyophilization processes, optimizing the freeze drying cycles for the novel peptide molecules. Why peptide we are discussing? Because the current generation is with respect to peptides, monoclonal antibodies, and the next level molecules. So here we have, because all these are most expensive molecules, so we have to use all available artificial intelligence to develop these particular molecules well. And the third is the regulatory filings, right? CTD preparations for the complex molecules, because there is a lot of phase 1, phase 2, phase 3 trials, and there are a lot of scale up trials, lot of CQAs, CPPs, how these we are controlling, and what is the stability protocols, stability data, post approvals, how we are going to launch everything. A lot of things will be there. So all these should be documented properly, arranged properly, then only we could be able to retrieve it properly. Computer vision in injectables, as I told you initially, if any vial fillings are happening, so generally in the past and olden days, humans used to see how whether the filling is accurate and any particles are there or anything. So wherein the always the speed is questioned, because humans they can become a fatigue, so they could not able to continue give their contribution consistently across the time. right? Across the AI, it is not like that. It's there is no fatigue, no nothing. Once you kept the sensor, it will run consistently. Every time it will consistently give the one output. What is the advantage of this? So industry can deliver a, a quality product consistently and across the markets. So that's a great benefit, and with this the productivity is increased drastically. Challenges, yeah, we have seen till now what are the AI softwares, what are the industry challenges, how we can integrate it, and what are the improvements, case studies, and etc. But again, each and every concept there will be positives and negatives. Let us see the barriers, challenges and barriers for the AI impact. And so first thing, we this has been categorized into low impact, high impact, easy to solve, and hard to solve. This is called as a four blocker classification. So for example, here we can see that major barriers and long-term focus. For example, GXP validations of the AI models, ICHQ12, and multi-site AI model governance, FDR, EMA real-time release and acceptance, patient data privacy. So all these are, you know, it's a long-term problem and major barriers, because once the patient data has been given to the AI, we don't know how these AIs can utilize it further. So these are the major barriers. And the low priority and monitoring type of the things are these are legacy system compatibility, because today I have brought some AI technology, for example, as I told you, Schrodinger, uh, because I don't have a computer infrastructure which could able to adjust to the new softwares, right? So this is what uh, how hence we have to monitor how to do that, and as well as the major workflow disruptions and vendor lock-in risk, because any new thing comes, vendors always will say that hey, you have to be with me for at least 2 weeks, 2 years, 3 years, and etc etc. Okay, so all these things to be monitored, and non-critical process automations. Quick to win is data quality, as everyone awares that, for example, if something done by a calculator, simple calculator, that will give you accurate than by the humans. That's what it's always quick to win and address fast, right? A literacy training programs, LIMS integrations, because stability data, everything it will integrate across the 100 batches, and it will say the pH of the 100 batches is within the limit. Very accurately it will say, along with the graphical presentations. And as well as the pilot project infrastructures, it is very easy, it will quickly draw and give to you, right? And the strategic priorities and plan now is regulatory acceptance of AI submissions. At present, I told you the wallets, which are at now a human interface is loading everything, but in future, once the development data is done, automatically it can convert it into the AA submission patterns. That's a strategic portfolio, and we have to plan now for this, right? And the IP, data ownership, and black box modeling, uh, organizational change management. So these are the few of the challenges and barriers. So what is a future roadmap? Injectables 2024 to 2030 plus. As we in the starting, we have told that we have a exponential growth in the AI. So this will integrate to that. So 2024 and 25, and pilot and proof of concepts most. So AI formulation screening tools deployed, and as well as the first NLP assisted CTD filings, PAT AI integration in clinical manufacturing. This is where generally at present the industries are looking, and later to this phase wise, it will goes to the scale up and standardizations and regulatory integrities and autonomous manufacturing. So in this way, uh, the journey follows from pilot adoption to the fully automation of pharmaceutical manufacturing. If you start in 2024-25, it will go by 2020 in fully automations across the all phases. So this is simply a beneficial comparison of AI traditional approaches, AI versus traditional approaches. As we learned by now, it it gives very quick, accurate data, either at with respect to dimension of formulation screening, stability predictions, and particle inspections, regulatory drafting, root cause analysis, supply chain plan management, literature mining, and as well as the process scale up. So there are a lot of advantages. So by learning this, we could be able to get a great future. As we have only 4 minutes left, I would like to focus with respect to career point of view. Um, so these are the few takeaway points. Um, so machine learning tools which are free. These are the tools which are available in database. By learning this, it would be very easy for the careers in the industry, and as well as the vendor trainings. And we also have a certifications, and there are a career roles. So any questions? We have 3 minutes left. So any questions for me?
Sir, good afternoon, sir.
Good afternoon.
Sir, my question is just now discussed about various types of AI tools. Is Schrodinger or any other AI tool is helping that with respect to the drug dose, the excipients concentration, how much can be fixed or how much can be used, so that we can get optimized the formulation like that, sir.
Okay. Um, Actually, the short answer is the Schrodinger does not directly calculate or recommend drug dose. Okay,
Okay.
but it does help in the upstream steps of drug design that eventually supports you for the dose calculation. Because
Okay. Okay.
the Schrodinger can help you primarily for physics-based molecular modeling and drug design, right?
Mm.
Which means
Okay.
that predicting binding efficacies, optimizing drug target interactions, and
Mm.
designing better molecules. It supports for the early drug discovery, not for the clinical dose calculations.
Okay. Okay. Other than literature review, so how much amount of like excipient A, B, C can be taken for this particular dose of a drug? Like that, is it any tool is helping? Is any AI tool or Schrodinger AI tool is helping in that way, sir? I'm asking.
Yeah, so so in phase wise we have to see in the Schrodinger you will perform the pre-formulation studies, wherein,
Mm.
for example, the solubility we have to basically
Purple.
we should know the solubility. There you will bring the amount quantity of the solubility agents,
Uh,
and as the co-solvents can be defined over. There's a step one,
yes.
and then we have to use a next level DOE design softwares for optimizing, such as the SAS or
Mm.
informatics. Okay, there
Okay.
we will
Okay.
optimize the formulation. Now questioning to your answer finally, that
Mm.
there is a software called as a Phoenix, right? There there it will let you know the it will simulate the pharmacokinetic model. Okay, how much
Okay.
absorption, distribution, and metabolism. So with
Okay.
that, whether you have to inject a this particular drug as a 0.5 ml, 1 ml, or 2 ml, everything can be optimized by a Phoenix software. So
Ah, okay.
you solubility was designed by Schrodinger.
Mm. Yes, sir.
SAS optimized your formulation, Phoenix
Ah,
for ADME and defining the volume of the product.
okay.
Am I answered your question?
Yes, sir. Yes, sir. Thank you, sir. Thank you.
Thank you. Any other questions for me? Okay.
Sir, one more question, sir.
Mhm.
From my is a Schrodinger AI tool or software is helping not only this pre-formulation like a solubility, even for saturation solubility also can be predicted easily. Saturation solubility of a drug can be predicted.
Yeah, you see, basically you started with a normal solubility study, right?
Yes, sir.
So obviously, to a very short answer is yes.
Okay.
Schrodinger does helps with predicting solubility,
Okay, sir. Okay.
the normal solubility, and as well as including the saturation solubility, because it works on the, as I told you, physics based and machine
Now,
learning assistant
learning.
computation tool.
Yes, yes.
That's the main logic of this particular software. Hence, again, yes, the answer is yes.
Okay, sir. Thank you, sir. Thank you.
Yes. Yeah, if no more questions, this is my email ID. So anybody wants to reach out me, they can mail on this, so I could able to answer those questions in a defined time frame. Thank you all once again. I thanks to Acharya Nagarjuna University management, to principal madam, to all senior leadership of the Acharya Nagarjuna University College of Pharmaceutical Science. I really thanks to each and everyone for giving this opportunity. I feel proud to sharing my knowledge, what I do day in, day out in my organization, companies, and that knowledge, and based on the literature, to all the knowledge seeking audience of this particular presentation. Thank you. Thank you all.
Thank you very much, sir, for that wonderful and insightful talk. Your valuable words have truly enlightened us and provided us with a deeper understanding of the topic. We are grateful for the time and knowledge you have shared with us there. Thank you very much, sir.
Thank you.
Thank you very much, sir, for accepting our invitation. Behalf of University College of Pharmaceutical Sciences, Acharya Nagarjuna University, I would like to express my deep sense of gratitude to you, sir. Thank you, sir. We will see you in the future in our upcoming FTP, sir. Thank you very much, sir.
Thank you. Thank you all.
Uh, Dear participants, that is the end of today's session. Tomorrow, the sessions will start at 10 a.m. 10 to 11 a.m. one session by Dr. Prajapati sir, and 11 to 12 p.m. Dr. Suvarna Enduri ma'am, and 12 p.m. onwards we have online test and valedictory. Don't forget, test is valuable. So certificate will be awarded after getting 60% of the marks from the test and 80% of the attendance. Okay, so today afternoon no session is there, only morning sessions. First session 10 to 11 a.m. Second session 11 a.m. to 12 p.m. Third session is valedictory. So each and every candidate, those who are connected in the online, they must be present in the offline. The certificates will be awarded on the stage. If possible, all are come to ANU CPS campus to receive the certificate and kickback. Okay, so some exceptional cases is there. Otherwise, so mostly you have to come to ANU CPS to receive the certificate on the stage. Thank you very much, dear participants. Thank you all the resource persons. Thank you, my dear colleagues. Thank you very much.